Good morning, afternoon, and evening. To provide an opportunity for everyone to log in and connect to the GoToWebinar platform, I'm happy to present a brief video introducing DSX, the next generation of DILIsym software.
DILIsym Services, a Simulations Plus company, provides powerful science and innovative technologies to support research and regulatory interactions. DILIsym version 10, also known as DSX, is quantitative systems toxicology software capable of predicting and explaining drug-induced liver injury. This version offers a brand new user-friendly look and feel. DSX is designed to provide an indication of the potential DILI hazard posed by individual molecules and to provide deeper insight into mechanisms responsible for observed DILI responses at various stages of the drug development process. Predict hepatotoxicity for phase 2, 3, or 4 clinical trials in patients, first-in-human clinical trials in healthy volunteers, small and large preclinical species, and inter-individual variability in populations. What's new in DSX? Contact us today to save time and resources on DILI hazard research. Special programs are available for instructors, students, and researchers. Learn more at simulations-plus.com/DILIsym.
Wow, I am excited, and I hope you are as well. Welcome to today's webinar based on the Toxicological Sciences publication, Mechanistic Investigations Support Liver Safety of Ubrogepant, presented by co-authors Paul B. Watkins and Jeff Woodhead. A couple of housekeeping notes. For optimal video and audio connections, we recommend you close out any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. This webinar is being recorded for future playback on our website and YouTube channel. And for the Q&A session, you may ask questions anytime via the questions panel on your dashboard. You can, again, submit at any time. We strive to have at least 10 minutes of Q&A, so please don't be shy. We know there are lots of ways to spend your time, and on behalf of everyone on the DILIsym Services team, we appreciate you spending the next 60 minutes with us. Now, to get started, we have a quick poll question to tell us a little about yourself. How familiar are you with DILIsym software? We'll give you just a few seconds here. And we're going to go ahead and close the poll now. So we've got a fair amount of not familiar at all and brand new to DILIsym Services. So welcome to today's webinar. Now it is my pleasure to introduce today's moderator, Brett Howe, President of DILIsym Services. Take it away, Brett.
Thank you, Arlene, and welcome to everyone. We appreciate you attending today. I'm really excited to introduce our distinguished speakers. I think they have a great presentation and a really important application of DILIsym to share that's been published in Toxicological Sciences recently. So let me start by introducing Dr. Paul Watkins. He's a physician and the director of the Institute for Drug Safety Sciences and also the Howard Q. Distinguished Professor at the University of North Carolina at Chapel Hill. He serves as co-chair of the steering committee for the U.S. Drug-Induced Liver Injury Network. He's also the recipient of numerous honors and awards, including the Rawls-Palmer Award for Progress in Medicine from the American Society for Clinical Pharmacology and Therapeutics, and the Career Award from the Toxicology section of the American Society of Pharmacology and Experimental Therapeutics, among others that we don't have time to list today. Second, I'll introduce our other speaker, Dr. Jeff Woodhead. He did his training in chemical engineering and joined DILIsym Services about 10 years ago, at the early days. Jeff has been a key part of many DILIsym application projects and has authored many of the key DILIsym application papers, including manuscripts on numerous antibiotics, tolvaptan, lixivaptan, N-acetylcysteine use with acetaminophen, and of course, Ubrogepant. So with that, I'd like to turn it over to Dr. Watkins and Dr. Woodhead. Go ahead, Paul.
Okay, so you see my slides, Brett? What's up?
Yes.
Okay. So as Brett said, I chair the Scientific Advisory Board for the DILIsym initiative. I'm compensated for that. I no longer have any financial stake, in the financial success of the company. Actually, in that advertisement, I was looking in the corner right to see where you could click in 5 seconds to avoid the advertisement. And of course, Jeff Woodhead is an employee of the company. And by the way, I'm not getting any compensation for this talk today. So the outline of my talk is going to be drug-induced liver injury, or DILI, in drug development, DILIsym, a little bit about the background, and then the Ubrogepant story, predominantly from the clinical side, and then Jeff will take over. So this is not a surprise to anybody, I'm sure, watching this, is the tremendous attrition that compounds go through from high throughput screening all the way to eventually getting approval. And obviously, safety is a major reason why drugs fall out of development, really at every stage. And this was a summary of the experience of AstraZeneca and why drugs fail preclinically, phase 1, phase 2A, phase 2B. They didn't go to phase 3 because there weren't enough compounds to make it significant. But what you can see, just based on the shading, the tan, that safety is a major reason why drugs fail to progress beyond preclinical. And then in phase 1, it's still the major reason. Of course, that's not a surprise. You're dose escalating to look for safety. And then in phase 2A and 2B, it becomes less and less and less in phase 3 as well. And so people have said that, once you're in clinical development, the reason why drugs fail predominantly is they don't work or work well enough. But you could also say, well, what if you gave 10 times the dose? Would they have failed then for efficacy or safety? So obviously, I think safety, you can argue, but safety is still a major reason why drugs fail, even late in clinical development. And then if you look at the reasons for termination of drug development programs due to safety by organ system, this was again the AstraZeneca experience, but it, I think, has been generally true. You can see that, and here the preclinicals in red and clinical where the termination occurred. You can see that cardiovascular remains the major reason. Of course, that's and the preclinical is not very good because the clinical percent failure is higher than the preclinical. But that's, of course, a huge category. It includes prolonged QT, includes cardiomyopathies, includes vascular injury, atherosclerosis. But number 2 is liver, and you can see that there are surprises that occur after preclinical development and clinical programs are terminated. I just point out the kidney is the other major organ that preclinical doesn't do a great job of preventing clinical toxicity. And as you know, DILIsym has gone on now with actually getting large phase 1, phase 2 SBIR grant to begin looking at kidney toxicity, starting at the proximal tubule. So, I ask, is hepatotoxicity still a problem in drug development? I think there would be people at the FDA that would say it's no longer a problem, at least for them, because no drugs have been approved in the last decade, and it's actually a decade and a half, that have been withdrawn from the market in the US due to liver safety concerns. And that's because I think regulators and of course drug the developers are better able to define liver safety signals in clinical trials. And so the Hy's law case, the sign of liver dysfunction as well as abnormality in liver chemistries, has become sort of the gold standard and has been pretty good at keeping surprises out of post-marketing. However, the cost has been that clinical trials can be much bigger and longer to establish, to fully describe liver safety or establish liver safety. And that's because once you have ALT elevations, abnormalities in liver chemistries, you're really thrown into proving you don't have a problem or proving a negative, which we know can be very difficult. And the FDA has this rule of 3 things. So you need 3,000 patients to exclude an event, 1 in 1,000. So trials can get very big if liver safety is a concern. So an example is the direct-acting oral anticoagulants, or DOACs. The first in class was zimelagatrin that ended up being withdrawn from worldwide markets due to liver toxicity, this rare delayed toxicity I'll talk about called idiosyncratic hepatotoxicity. The next in class, rivaroxaban, was approved for chronic treatment in atrial fibrillation, but only after more than 35,000 patients had actually been exposed to the drug. And I think it's quite clear that that a lot of the desire for prolonged, huge exposure, almost post-marketing exposure for this drug, was related to the liver safety concern of first in class, even though rivaroxaban actually has a different target and very different structure. But that has been a pattern, I would say, at the FDA of if there's a liver safety concern with first in class, to have special scrutiny at next in class and heightened concern of liver safety. And that's relevant, as I'll show you, for Ubrogepant. And this was a little over a year ago. The company Motif Bio had an antibiotic for bacterial skin infections, and the complete response letter cited the need for additional data to assess liver safety. The company plans to meet with the FDA as soon as possible to clarify the path forward. It will need to raise additional capital in the near term since it's only financed into the next quarter. And tragically, the shares of the company went down 80%. And as far as I know, there's been no movement forward with this drug. And of course, the costs of additional studies are very substantial. But the economics is actually not just related to the cost of doing the study. And that's that if a drug attains at least $1 billion in sales at the end of patent life, so 365 days in the year, that's more than $2 million a day that's at stake for every day you don't get approval. So in terms of investors investing in drugs that suddenly are asked to do another large prolonged clinical trial to assess liver safety, it's a hard pill to swallow. And it's no wonder, by the way, that when drugs show liver safety concerns at any point in development, there's serious considerations of do you continue to develop that drug, even if there's an important unmet medical need, at least potentially. So hepatotoxicity remains a major problem in drug development, and current preclinical testing has not eliminated this problem. So DILIsym. DILIsym arose from the Institute for Drug Safety Sciences, which I established over a decade ago at the university, with the idea of studying rare or idiosyncratic but severe toxicities in patients. And as a hepatologist with a long-standing interest in drug-induced liver injury and liver, we focused on that primarily. And I've been heavily involved in the U.S. Drug-Induced Liver Injury Network, where we find these people and study them. Also collaborating with drug companies and interested in new biomarkers and things. And we established a variety of in vitro assays and cell culture systems, and mouse genetics has been a big part. But very early on, I realized the power of modeling, particularly what's called quantitative systems toxicology modeling. And over the last decade, I believe it's 17 or 18 of the top 20 pharmaceutical companies have participated in the development of the software, of this modeling software called DILIsym. And the FDA is, until recently, a year and a half ago, was funding a ORES fellow actually embedded in the team. The first ORES fellow actually went back and got a full-time job at the pharmacometrics division at the FDA. So there was great interest in developing this quantitative systems toxicology model that would improve understanding and predict liver safety liabilities in new drug candidates. My major interest actually was the experience I had with a company called Entelos. One of the wonderful things about this modeling is as you do it, it becomes clear where the gaps in knowledge are. So it really it prioritizes research really for the entire institute, which it did. So quantitative systems toxicology is the use of differential mathematical equations to recapitulate relevant pathways whereby drugs or other chemicals can cause stress and death to cells, tissues, and organs. And the approach has been within the DILIsym initiative to build these mechanistic modules using differential equations, such as glutathione homeostasis, things that are relevant or soon to be relevant for a drug-induced liver injury. And then as gaps are identified, fill in those knowledge gaps with research. And then integrate these modules with the outcome of hepatocyte death and release of its contents, which includes alanine aminotransferase, as well as functional impairment with bilirubin. Based on the traditional clearance, release and clearance kinetics. And that's included now novel biomarkers such as miR-122, GLDH. And then in the model to vary the parameters to create a simulated patient population. In other words, vary the parameters based as much as possible on literature, for instance, variation, expression, and activity of BSEP, bile salt excretory protein, to reflect the genetic and non-genetic factors that create heterogeneity and susceptibility to DILI. And then over the decade, the model has been refined by incorporating data obtained from successive exemplar drugs. So these are drugs where some information is available in literature, but generally compounds that were provided by the partners where there was a discrepancy between preclinical and clinical safety. And that's been refined, and now there's a validation cohort, et cetera, that the model's being tested on with about 80% predictive in terms of toxic or non-toxic. And we can talk about that in the questions. This is the 30,000-foot view of the different modules that are involved in hepatocyte injury. There's another branch that's actually cholangiocyte for cholestatic toxicity. And you can see there's some of the things you'd expect. There's drug metabolism, distribution, PBPK. There's reactive metabolites. There's reactive oxygen species, mitochondrial toxicity, alterations in intracellular bile acid concentrations. Lipotoxicity is a more rare cause. And they all feed in together, with the result being hepatocyte death and release then of ALT, which is contained in liver cells as a function of time, actually graphs it, it's a function of time. But also as liver cells volume drops, there's regeneration built into the model. And innate immune responses to the injury and also participating in the injury are built into the model. And one of the surprises that came out of the modeling was that just 3 properties appear to account for the vast majority, again about 80% in the validation cohort, of dose-dependent liver toxicity, and also accounts for largely species differences in dose-dependent hepatotoxicity. And that's reactive oxygen species, or oxidative stress, mitochondrial dysfunction, or alterations in bile acid homeostasis, again impacting liver cell death and release of these biomarkers into serum. Now the way this works then, and alanine aminotransferase is what I'm going to be talking about. is you need to have some estimate or prediction of what the exposure is to the liver, both outside and inside the liver cell, the hepatocyte. Then in a laboratory and commercially available, you can measure the ability of the drug and major metabolites to inhibit function of bile acid transporters. You can also study the effect of the drugs and metabolites on mitochondrial respiration and generation of reactive oxygen species. Done by and large in HepG2 cells, but now the initiative is moving towards metabolically competent cells such as HepRG, but also spheroids and more complex organoids, mainly to account for metabolites, unknown metabolites in the process. And then again, interpatient variability, such that when you put in this information, outcomes without manipulation, true prediction, the incidence and severity of elevations in alanine aminotransferase as a reflection of liver cell death. So, two broad types of hepatotoxicity. There's the direct or intrinsic toxicity, and then there's the more problematic idiosyncratic toxicity. So, in the direct or intrinsic hepatotoxicity, this is clearly dose dependent. Now, there can be large differences between people in susceptibility, but if people take enough of direct toxins, the idea is they would all get the toxicity. So acetaminophen is the classic example. Typically has a short latency, within the first week of treatment. It's usually, but not always, evident in animals, and there are DILIsym publications showing variations, certainly with bile acids, where the profile is less toxic, generally in animals, but also differences in species and interference with mitochondria function. Again, usually identified at the level of phase 1 trials with dose escalation. And DILIsym has had a very high predictive run at this type of toxicity. The more problematic, though, is what's called idiosyncratic hepatotoxicity. And here the dose relationship is complex. People have said if you get less than 10 milligram of a drug a day, you shouldn't have a problem. So there's some threshold in terms of that needs to be crossed, but it's a complex relationship. The latency is longer, typically 1 to 6 months after starting treatment. There are no good animal models for this latency and idiosyncratic nature. And unfortunately, it's usually identified late in clinical trials or post-marketing. And again, the idea is the drug is safe for the vast majority of people who take it, the green part of the triangle, but there's a subset that can have toxicity, and those that take it safely may not even have the slightest flicker of liver stress, that there's something very different about a subset with these drugs, that a subset of patients that makes them vulnerable. And we now know that in many cases, and perhaps most cases, this event is caused by an immune attack on the liver. This is the course of a patient that developed tolvaptan hepatotoxicity. And what's shown are the various liver chemistries, the traditional ones. I'll concentrate on ALT here, which is the black triangles. And this is the time on drug is on the x-axis for this fellow. So they started, and you can see the increased shading. There was dose titration up to 90 milligrams and 30 milligrams a day. And then at around 4 months, the ALT started to rise. 5 months, it had gone over 3 times the upper limit of normal. The drug was stopped, but in spite of stopping drug, and by all accounts, all the drug being out of the body, the liver toxicity raged on for weeks before finally turning the corner and coming back down. At this stage, it wasn't sure there was tolvaptan. They started again at the lowest dose, and within the first week or so, first blood drawing, the toxicity was back. So memory going on when the drug's gone. This is a classic adaptive immune response that was initiated by the drug. For some reason, the immune system thinks the liver is virally infected and starts trying to kill these virally infected cells. But there is no virus, it's the drug. So idiosyncratic DILI usually involves an adaptive immune attack on the liver. So does DILIsym predict this liability in new drug candidates? I think the first reaction would be no. If this is an adaptive immune attack, and although in the next phase and ongoing right now is bringing in adaptive immunity, T cell kinetics, et cetera, into the model, the current model and predictions have not involved that. So does DILIsym predict this liability in new candidates? And the answer appears to be yes. The first manuscript in 2014 really reproduced very closely the toxicity of troglitazone, even the latency to the peak ALT value was about 4 months, 4 to 5 months, which is when the toxicity occurred, including the severe idiosyncratic toxicity with this drug. And the idea is something's going on. You don't need 5 months to develop an adaptive immune attack. Obviously, and so this prediction that's in DILIsym appeared to be accounting for some of this delay and being able to account for the toxicity based on direct toxicity modeling. Again, with TAC-875, this was a drug that died late in phase 3 dosing. And then tolvaptan, I gave you the example of that. Again, DILIsym picked up these drugs as having a liver safety liability, even though the toxicity that was of concern was clearly this delayed, sudden, severe liver injury. So why would that be? Well, again, if you have a drug exposure, if the adaptive immune attack is the final step that leads to liver injury, we know that there are other things required for an adaptive immune attack. Even to develop immunity with a vaccine, you need adjuvants. And I was told the first adjuvant was actually turpentine. You need something to create inflammation, the danger signals. Just having a novel antigen is not enough. The adaptive immune system must also perceive that there is a pathology occurring. So you need, we believe, hepatocyte stress. This is what, and perhaps death, but at least stress. You need neoantigens. The hepatocyte stress is obviously clearly in DILIsym. You then need release of these damage-associated molecular patterns or DAMPs, and you need this innate immune response. You need the local inflammation. This is the equivalent of the adjuvant effect. And this really is the sweet spot for DILIsym. And the idea is if DILIsym says this doesn't occur with your drug at a dose at your planned dosing interval, this shouldn't occur. Obviously, in a subset, this is occurring presumably in everybody to lesser degrees, but you have to have the right HLA predisposition, maybe PTPN22, et cetera, a variety of immune susceptibility, genetic susceptibilities that are being identified in these large registries like the DILIsym network. Now that remains largely hypothesis. I don't want to oversell it, but I would say so far so good. So let me now go to the Ubrogepant story. So here's Ubrogepant, the Merck drug. It's a calcitonin gene-related peptide, CGRP antagonist for the treatment of migraines. They already knew that the monoclonal antibody was very effective and didn't cause toxicity. Apparently there's a knockout mouse, I think there's a knockout human. So that there was no on-target concern about hepatotoxicity with a small molecule. And as you can see in the bottom, there were pretty wide exposure margins in rat, both 3-month, 6-month, 9-month non-human primate, nothing at 7 times the margin, and then a 6-month mouse study, nothing really. So this looked good. However, when Merck did a randomized controlled trial of this drug to prevent migraine, giving 140 milligrams or 280 milligrams or placebo twice daily for 12 weeks, they saw serious liver safety signals. Interestingly, they also saw a dose-dependent increase in ALT elevations, again supporting that there is a dose, a relevant dose response in liver cells to this drug. But there were 2 cases of severe liver injury, and this is a 25-year-old woman 2 weeks of treatment with telcagepant, 140 milligrams. Her laboratory results were within normal limits, but at 4 weeks, her ALT had gone to almost 40 times the upper limit of normal. She complained of abdominal pain and nausea. So this is a severe symptomatic liver injury that was delayed latency. The other patient was a 45-year-old woman, 2 weeks of treatment with telcagepant, 280 milligrams. Her ALT level was only slightly elevated at 2 weeks. She experienced severe flu-like symptoms days 17 through 24 with nausea, anorexia, dyspepsia. And then at 4 weeks, she was noted to have these very high transaminases. So these were two symptomatic severe liver injuries that really made continuing to develop this as a prevention unlikely. So Merck then said, well, since nothing really happened before 2 weeks, why don't we go for a treatment of migraines associated with menstruation? So they did a large study, phase 3, of 2,660 patients, got the lower dose for 7 days each month versus it was a 2-to-1 randomization of placebo. However, there were 3 patients, all in the telcagepant group, that had ALT elevations greater than 8 times the upper limit of normal that were considered a serious liver adverse event. So this, Merck decided not to pursue this drug any further. And obviously this hurts if you do a phase 3 study, this deep into the clinical development to scrap it. So they went on to their next in class, which is MK3207. The structure is similar, certainly in the business end, which is on the right. Again, great safety margins in rat, non-human primates, mouse, dog. However, and this hasn't been published, this was abandoned earlier in clinical trials because of liver safety concerns. And actually, according to the publications, these were short-term treatments initially. The liver injury occurred after they came off drug, clearly indicating to me an adaptive immune mechanism for this drug. So they had a third in class, Ubrogepant, which did a little better in terms of safety margins in the animals. But Merck had enough. They decided that they didn't want to had higher priorities probably for development, and sold this drug to Allergan. So with that, I'm going to turn this over to Jeff, who will tell me when to advance the slides for how this issue was approached by DILIsym.
All right, thanks, Paul. So as Paul alluded to, we were brought in to take a look at Ubrogepant with the DILIsym software and compare it to telcagepant and MK3207. And so what we did is kind of the standard DILIsym way of doing things is we take each of these compounds and we have in vitro experiments performed for each of the, as Paul mentioned, there's 3 main DILI mechanisms that we are able to understand and predict. Mitochondrial dysfunction using the Seahorse XF analyzer. Bile acid transporter inhibition using kind of vesicle assays that I think are fairly common in the industry nowadays. And oxidative stress, which is a high content screening with DHE. And those are all generally performed by contractors. So we send compounds to them, they send us results, and we perform simulations with them along with PK models for each of the compounds. So in this case, what we were looking at was we have 3 different compounds, 2 of which we knew what we should have been getting, right? We wanted to be able to predict telcagepant and MK3207, this toxicity that was seen in the clinic. We wanted to recapitulate that clinically observed toxicity. And then when we go forward with Ubrogepant, our goal was to predict the likelihood of toxicity using the same simulation method. So yeah, everything here is a pure prediction, obviously. There's no, the clinical data doesn't get fed in, but it's useful as a comparator to make sure that we're on the right track. Because if we predict Ubrogepant is clean, but also predict telcagepant is clean, well, you can't really doesn't really say a whole lot. So it's important to make sure that we were on the right track with being able to predict telcagepant and MK3207 before we look at the meaning of the Ubrogepant data. So if you go to the next slide. So this is just kind of how everything comes together in DILIsym. And Paul alluded to this earlier, but we, specifically what we do is we take some of the various compound properties and whatever data we have from the clinic, whether it's pharmacokinetic data, some preclinical data about tissue distributions, etc. We build a PBPK model that predicts the plasma and liver concentration for each compound. On top of that, we layer this in vitro mechanistic DILI data that I mentioned earlier, the oxidative stress data, the mitochondrial tox data from the seahorse, and the bile acid transporter inhibition data from vesicle studies. And we use those to produce a representation of each compound. We run the simulation. We will the outputs of that are certain biomarkers and certain the ALT, bilirubin, etc. And we then compare those to what we saw in the clinic to be able to assess whether or not we have an inaccurate representation of the compound and whether or not we're really on the right track here. So you go to the next slide. So what we wanted to do is we wanted to look at, for MK3207 and for telcagepant, looking at the clinical doses at which toxicity was observed. For MK3207, it was these 200, 300, and 450 milligram dosing levels, where they gave 2 doses, oddly enough, 2 hours apart. For 2 weeks. And this was kind of an earlier in development. I think it was phase 1, if I'm not mistaken. It may have been 2A. But it was a fairly small end study. And at those dose levels, what we knew is that a fairly significant percentage of individuals were getting toxicity. And most of those responding were at the 600 and 900 per day levels. Paul already showed the telcagepant data that was at 140 BID and 280 BID for 12 weeks. There was less than 140 and then more at 280. And then, of course, we go to Ubrogepant. We had these proposed clinical dosing protocols. We knew it was a treatment for acute migraine. So we looked at 8 days straight of 100 milligrams QD. And then split 100 milligram dosing for 4 days, and then this kind of what we expected to be the maximum dosing scenario, which is this 2 days on, 2 days off, 100 milligrams for 56 days total. So that would mean 28 doses over 56 days. And so, of course, we didn't know what those simulations would produce at the time of the simulation. So if you go to the next slide, this is a little bit of a demonstration of the in vitro data we get. This is for telcagepant. You can see here that on the left, these are the data from the XF analyzer demonstrating some mitochondrial effects that telcagepant is very clearly inhibiting the electron transport chain. You can see that as you increase the telcagepant concentration, the mitochondria stop consuming as much oxygen, which suggests that some of those ETC proteins are inhibited by telcagepant. On the right here, you see oxidative stress, where increases in telcagepant concentrations in the cell are associated with increases in measured RNS-ROS. And so what we're able to do is we're able to take those data and translate them into a number. And that number represents how potent the molecule is for that particular mechanism. So we have a coefficient for electron transport chain inhibition and RNS-ROS production rate constant. And if you go to the next slide, you'll see that we also look at BSEP inhibition, and we get a number from that as well. That number is a little bit easier to come by because it's generally just a KI. In this case, we did a full KI study, which involves looking at your typical IC50 study over several different substrate concentrations, so we can get a sense of what the mechanism of inhibition, what the mode of inhibition is for this compound. It turns out that's quite important. We've seen a few compounds where if you model it as a competitive inhibitor, it models clean. If you model it as a non-competitive inhibitor, you see toxicity basically everywhere. And telcagepant was one of those where we needed to get those data. So we ran the full KI study through our contractor. And it turns out that both Vmax and Km were affected by telcagepant, which suggests that we had a mixed style inhibitor. So we got a Ki and this alpha value here for mixed inhibition with telcagepant, and so that gives us a full representation of telcagepant in DILIsym. Each of those three mechanisms now has a number that we can input into the software to produce a prediction of whether this compound is going to hurt people's livers. If you go to the next slide, you'll see that we did that same thing for each of the three compounds that we're looking at here. We looked at telcagepant, we looked at MK3207, we looked at Ubrogepant, and we found some signals for each of those compounds in each of the three mechanisms. And so, we took these numbers, we combined them with the PK model. These numbers don't really mean a whole lot on their own. They're inputs into the software. They're just the numbers we use to parameterize each of these compounds. And so, you can look at each of them has a little bit of oxidative stress. Each of them is producing a little bit of ETC inhibition. Each of them is inhibiting BSEP a little bit. And so now we're going to see what affects each of those mechanistic assay results has on our predictions of toxicity. So if you go to the next slide, you'll see the simulation results here in this column. What did the software predict? For MK3207, we found that there was some toxicity, in fact, significant frequency of toxicity at each of the doses, the 200, 300, and 450 milligram Q2H, 2 daily dose protocol for each of those simulated doses. So, and you look at telcagepant, we predicted nothing at the 140 milligram dose. We predicted a substantial level of toxicity at the 280 milligram dose, suggesting that if we're doing pretty good here in terms of believability, right? We're predicting some toxicity for telcagepant and for MK3207. Check those boxes. So we know that basically whatever we predict for Ubrogepant, we can kind of trust that's going to be a fairly trustworthy prediction, because we've validated those predictions with those other two drugs in the class. So we go to Ubrogepant. We've simulated each of these dosing protocols, these proposed dosing protocols, and it was completely clean. No individuals had significant ALT elevations. There was just a whole lot of nothing going on in those simulations. And so that kind of gives you this idea that, well, now the simulations are telling you that Ubrogepant is very clearly safer than MK3207 and telcagepant, at least with respect to these mechanisms that we have in DILIsym. And because those mechanisms are predicting what we saw in the clinic for telcagepant and MK3207, you can go forward with a little bit of confidence saying, all right, Ubrogepant is probably pretty certainly more likely to be safe than those two earlier in class drugs. If you go to the next slide, these are the EDISH plots. It's a kind of a format that the FDA often likes to see data in. On the x-axis here, we're looking at the peak ALT in terms of fold above upper limit of normal. On the y-axis here, we're looking at peak bilirubin, also in terms of fold above the upper limit of normal log-log graph. And what you can divide these graphs up into these quadrants, where the bottom left is normal range, not a whole lot going on. You're in the clear. The bottom right here is the Temple's corollary range, where you have an ALT increase without a bilirubin increase, where kind of look at that as a flashing yellow light. You're starting to cause some problems. Up here in the top right is your Hy's Law range, where you have both ALT and bilirubin increases. That is big flashing red light. Don't go here. If you have individuals in this, they're likely to develop serious hepatotoxicity, and that's a problem. And you can look here in the simulation. These are simulated results. For MK3207 and for telcagepant, we both had individuals in that Hy's Law range, and quite a few individuals in that Temple's corollary range. That there's some pretty significant and quite severe toxicity simulated by DILIsym, which tracks somewhat with what we saw in the clinic, right? There were some pretty serious events going on in the clinic for both of these compounds. But if you look at the diagram that we simulated for Ubrogepant, I mean, that's pretty clearly different from what you're seeing over here, right? Every individual is in this normal range, and it just looks like a single dot because all those dots, every 285 individual in our population is layered right there on that single dot. So you can see that not only is there just a lot less going on, there's really nothing going on in that simulation. And so that's just kind of adds to the weight of evidence that suggests that Ubrogepant should be safer than those other two drugs. So if you go to the next slide and kind of wrap up what we did with the simulation project here, that we predicted the liver toxicity for telcagepant and MK3207, fairly consistent with the clinical experience. So we check that box. We have a reasonable simulation paradigm here, and then prospectively predicted that Ubrogepant would be safe in the clinic. And that was what we did here at DILIsym. So Paul, I think you had a couple of other slides to take over.
So here we go. So DILIsym predicted safety liabilities for telcagepant and MK3207. And just to reiterate, these are predictions. It wasn't, a lot of times with modeling, people say that they were able to predict. Well, they meant they tweaked the variables to get the answer that they wanted. This was, the current DILIsym version, putting the data in, pressing the key on the computer and seeing what it spits out. Whereas Ubrogepant was predicted to be safer. And sure enough, it turned out to be safer in clinical trials. This is a result of a publication at the end of last year, where Ubrogepant was given 100 milligrams on 2 consecutive days, followed by 2 days of placebo. So this is, going for an extreme dosing of acute migraines. And what you can see down at the bottom is that elevations after, baseline on ALT greater than 3 times the upper limit of normal, which is a traditional cutoff, was not any different between Ubrogepant and placebo. Interestingly, there was one case, this is in the publication, and only one case where the ALT elevations were judged to probably be due to study drug when it was blinded whether it was study drug. It turned out to be on Ubrogepant. This was, just an ALT elevation, asymptomatic, but it's interesting, it was in the Ubrogepant category. But the Food and Drug Administration at the end of last year approved Ubrogepant for the acute treatment of migraine, with or without an aura in adults. And there's no safety warning in the package insert, and there was no advisory committee. And I don't know, I know that the DILIsym modeling was part of the NDA package, and I don't know, exactly, how much this may have been part of the decision, but I suspect it was part of the weight of evidence that gave confidence to the division to go ahead and approve this drug. So DILIsym modeling was part of the weight of evidence that supported FDA approval of Ubrogepant for the treatment of acute migraine headaches. It's interesting, back in 2014, when that troglitazone manuscript appeared in Clinical Pharmacology and Therapeutics, it was accompanied by a editorial from Schoen and the late Daryl Abernathy in the Office of Clinical Pharmacology, praising the approach, and you know, it was very nice to read this as one of the first publications to come out of the DILIsym initiative. But he also said, we look forward to future efforts to apply this model for prediction of hepatotoxicity that has not been observed clinically. Well, Ubrogepant is the first example where the modeling was done prior to human exposure. It was part of the weight of evidence that encouraged, I think, that led to the decision to go ahead and develop the drug. And there are now several other drugs that are in phase 3 clinical trials that had the same thing, next in class, where liver safety has been a concern earlier. And so far, so good. And you'll be hearing more about that, and hopefully will lead to publications as well as FDA approvals. So here's the team as it exists now, the DILIsym Services team, which is the umbrella organization for the public-private partnership, the initiative. And I would like to point out that recently Sergey Ermakov has come from industry to join the team full-time. And Sergey has really been a leader in industry in the whole field of quantitative systems pharmacology, and we're delighted to get him on board. I'd also like to recognize the outstanding scientific advisory board we've had, including the likes of Neil Kaplowitz, Kevin Park, Jack Utrick, Bob Roth, and David Pisetsky, and our new addition, Frank Sester, who left Merck and has been a leader while at Merck in preclinical toxicology and innovative approaches, biomarkers, et cetera, been involved in several major pre-competitive national and international efforts. We're delighted that he's joined our scientific advisory board. So with that, we'll quit and have about 10 minutes for questions.
Thank you, Jeff and Paul. This was very enlightening. Question for the audience. Your primary interest in either using DILIsym software or learning more about DILIsym software is what? You can choose all of the answers or none of the answers, multiple answers if you'd like. But please let us know how you would like to use and or are currently using DILIsym software. And we'll give you just a few seconds here. Thank you. And right before we begin our Q&A, based on what you've heard today, would you like us to contact you to begin a software evaluation of DILIsym? Go ahead and select your choice while Brett Howell prepares for the Q&A session. All right, I'm going to close the poll now. And if you have not already provided us a question through your questions panel, you can do so now. And Brett, I will turn it over to you to begin the Q&A session.
Okay, thank you, Arlene, and thanks again to Jeff and Paul for an excellent presentation. We have a number of good questions here. We'll start with Jeff. So this question is for you, although both of you, of course, free to comment on any of the questions. How do you define mitochondrial dysfunction, and what processes are considered within these, assays and modeling projects?
It's a very good question. So the Seahorse XF analyzer is really kind of put together to detect two main mechanisms that we see of mitochondrial toxicity. One is electron transport chain inhibition, where you see this decline in oxygen consumption as the compound concentration increases. Basically just means the ETC is not working as well as it could be. The other that we've seen from time to time in certain compounds is uncoupling of the mitochondrial proton gradient, where and that will have the opposite effect in the seahorse assay, where you'll see the oxygen consumption rate go up because now you're forcing the electron transport chain to work harder and harder and harder to keep the proton gradient at a level where it needs to be. At least at certain doses, and as you increase the concentration of the drug, you'll eventually see it just give out because it can't work any harder, and you'll start to lose activity. Those are two main mechanisms of mitochondrial dysfunction that we see. We'll see a couple of others here and there. For example, direct inhibition of the ATP production by the mitochondria. That's another one we can we'll be able to see with the seahorse. There's a couple of others that we've looked at, but really uncoupling and ETC inhibition are the main mechanisms that we see that drugs by which drugs affect the mitochondria.
Okay, thank you. Now let's go to Paul. Paul, did DILIsym modeling impact the number of patients required to be studied in the phase 3 study for Ubrogepant?
Yeah, that's a good question. I'm not sure. I wasn't involved in discussions of sample size. That was, it's certainly our hope that clean DILIsym modeling will lead to, more efficient drug development, smaller required trials, et cetera. But in the case of Ubrogepant, I don't know whether the modeling results had an influence on the design and size of the trials. I just don't know.
Okay, thank you. And Jeff, this one is for you. It's two different questions. So let's start with the first. It's a simple question. Is there anything special about the number of individuals in our SimPops?
So that's a good question. There's the number was really chosen because of the amount of parameters that we have, variability in. 285 is a good number to be able to reproduce the range of each of those parameters and get some combinatorics between each of those ranges occurring, while not crushing our computer under the weight of thousands of individuals, which would just take forever to simulate. So that area around between 250 and 300 ends up being a pretty good sweet spot for being able to limit our computational time and still represent the vast majority of the variability we see in certain parameters.
Okay, and a somewhat related question and probably our last, is a lack of patient variability a possible explanation for the overprediction of DILI liability for MK3207 and underprediction of DILI liability for telcagepant?
I think it's, possibility. I think there's also the possibility there's a little bit of uncertainty in some of those tox parameters. You can go, maybe a 10% up or down on that. I mean, if you see the telcagepant graph, you can see that there's a couple of error bars there that we could move up and down, and you could plausibly tweak that number a little bit and get a few individuals with tox at that 140 dose. I think there's a couple of other areas of uncertainty there that where the frequency of toxicity might be slightly different. There's also severity issues. There's a couple of adaptation mechanisms that we know happen that we're still getting a handle on in terms of representing those. So you saw with telcagepant simulated EDISH that there are those 20 or 30 individuals in the Hy's Law range that probably wouldn't have been there in the clinic. We didn't see that many Hy's Law cases in the clinic. So, so that could be a potential reason why we would overpredict, at least with severe, in terms of severity. I think we were there's also the question of, for MK3207, the clinical trial has had a fairly small N. It was 5 out of 12 individuals, and if you'd expanded that clinical N to 300 individuals, well, yeah, sure, it could have been 150. It also could have been still 5. So there's a pretty wide range that of potential outcomes for a larger population suggested by the outcome of the MK3207 clinical trial. So, and because I didn't go forward with it, obviously, I think we're actually pretty good in terms of frequency there.
All right, sounds great. All right, well, thank you guys. I'll turn it back to you, Arlene.
Thank you, Brett, Jeff, and Paul. Software evaluations are available by requesting them through our website at www.simulations-plus.com. This does conclude our webinar for today, but we plan to keep the conversation going through our LinkedIn page. We look forward to seeing you at the next webinar for the official DSX software release. Have a great day, everyone.
DILIsym Services, a Simulations Plus company, provides powerful science and innovative technologies to support research and regulatory interactions. DILIsym version 10, also known as DSX, is quantitative systems toxicology software capable of predicting and explaining drug-induced liver injury. This version offers a brand new user-friendly look and feel. DSX is designed to provide an indication of the potential DILI hazard posed by individual molecules and to provide deeper insight into mechanisms responsible for observed DILI responses at various stages of the drug development process. Predict hepatotoxicity for phase 2, 3, or 4 clinical trials in patients, first in human clinical trials in healthy volunteers, small and large preclinical species, and inter-individual variability in populations. What's new in DSX? Contact us today to save time and resources on DILI hazard research. Special programs are available for instructors, students, and researchers. Learn more at simulations-plus.com/DILIsym.